There's one more session now, one short one.
Okay, thanks everyone. So the next session, Klaus Wimmers from FBN is going to present the EuroFAANG research infrastructure. So research infrastructures are funded through Pillar 1, so a different pillar to the to projects like BOVREG and EuroFAANG and GeneSwitch. And their aim is to provide access and build capacity and consolidate research in the European research space rather than generating data per se. So over to you, Klaus.
Thank you, Emily, and thank you, Krista, for inviting me to present this project here. That is really, I think, of interest for the whole community of these FAANG projects that are ongoing. And I would like to first have a view on this time scale. And of course, this scale even began in the last millennium, and those that are already in animal genetics during the last millennium know all these steps from microsatellite markers to QTLs, EST mapping, then we had the SNPs, SNP mapping, and finally the genome sequences. And all these milestones really were the starting points for some advance in predicting the inheritance and the expression of the important traits. And when we had the genome sequences of the animals, then it was immediately clear we need to know more about the function of the genome than just the protein coding. And that was actually the start of the FAANG initiatives on the international level, and on the European level, it got the first funding and cost action, the FAANG Europe cost action. And later on, there was a funding of the independent FAANG projects that are, one of these is, of course, BOVREG, and others were presented today. And it is really a very good success that these projects, these individual projects succeeded in coming up with one EuroFAANG cluster. And this is, of course, due to the fact that these projects do have common research aim. They are all focusing on increasing efficiency through precision breeding. They aim for increasing disease resistance and to minimize environmental impact. And they do have joint strategies with regard to communication, training, and also the methods that are used. So this EuroFAANG cluster then evolved to the EuroFAANG RE projects that I'm talking about today. And this project, the European Infrastructure for Farmed Animal Genotype to Phenotype Research, is a Horizon Europe infrastructure concept development project. So this is funded in the InfraDev stream of Horizon Europe. It started last year for 3 years, and you see we have 7 partners which actually do represent the individual EuroFAANG projects, and the project is coordinated by Emily and me, and actually I took over from Krista when Krista moved from the FBN to FLEE. The aim of the project is to establish a concept for an infrastructure to facilitate research and innovation for G2P research in farmed animals. And to make it clear, we are a concept project, so we do not do research and we do not provide infrastructure access and services at this phase. So this is also different from ongoing infrastructure projects like PigFarm or SmartCo, your way also now. But we are aiming to bring together the infrastructures and resources here in Europe to get a plan and concept that is eligible for the S3 roadmap, to get a sustained support for the G2P research in farm animals. We started a large number of activities, surveys, workshops, and so on, to map what's present and to identify gaps and needs. And it is important that we really have a comprehensive collaboration within the whole community in order to fit the demands that are from stakeholders and from the science here in Europe. And there will be a number of opportunities for you to associate into our activities. It's wrong. Maybe just a few words about the ESRI environment. So ESRI stands for European Strategy Forum for Research Infrastructures. So this is an idea to develop roadmaps for research infrastructures to be funded in Europe. And in the area of health and food, there are actually 16 infrastructures, and 5 of these in the domain that is of interest for us. The domain food, it's on biological resources and production systems. But among these 5, there is none about farm animals. So there is a lack, there is no infrastructure at the moment for farm animal research. And also the free infrastructures in the field of biological resources that do have similar profile than that what we may have. It's on animal models, and human biobanking and microbial resources leave open a gap that is to fill. And these ESRI roadmaps are reviewed continuously and rewritten continuously. And in 2021, it was stated that there is a need for concerted efforts to continue bringing together national facility at a pan-European level in the field of animal genetic resources, phenotyping, breeding, and animal health to contribute to safe, healthy, and sustainable food supply. So you see there is really a lack, and I think we would really fit into this landscape of ESRI projects here. So what are our main objectives? Our objectives are the creation of common data structure and data access through the FBN data portal that's already running and in connection with the Elixir community. We want to develop a European framework for curation and biobanking and in vitro cellular models. We want to share and expand the capabilities in new breeding, phenotyping, and genomic methodologies. So genome editing is an important part. And we want to connect the existing projects and infrastructure really to foster the G2P research in Europe. We actually had our kick-off meeting just about 1 year ago in February in Berlin last year. And since most of the participants already know and collaborated, they are experts, we really had an immediate start into work in this project. And I will like to mention some of the activities that we had. We had, for example, training and capacity building activities. I mentioned here the summer school and JUI in last September about cellular models. Then what we have is we do have so-called think tanks on genome editing, on biobanking, and advanced research methods. And the first virtual meeting on the genome editing was in October last year. It was a really successful first online meeting where we were able to really point out the barriers, the opportunities, and the future priorities to foster these techniques. And this was supported by a survey which delivered a preliminary map of the labs that are actually doing GE research. And I guess that in future you might be addressed by other surveys from us. And I really like to ask you, please answer to this service. It is really helpful for us to come up with a final successful concept of the project. Our project was also presented at the EFAB Fabre meeting last May, and there we also already had also some world coffees here. It was also in this room where we discussed biobanking and genome editing. And we did, together with you, the EuroFAANG workshop at the EIP. I think that's important because it's also addresses stakeholders outside the research sites. We did the survey on biobanking, and we do have other surveys around. So at the moment, I think we already have quite a good picture on the activities, the facilities, and expertise in Europe, and where are the gaps and what are the challenges and opportunities to really build up EuroFAANG research infrastructure. You see, we plan some activities for today, for this year. One is the conference here today. Thanks for this opportunity. And we do have also plans within the project, and we also plan to have a white paper on in vitro systems for G2P research, which might be interesting and really outreach. So we can only be successful if we really are able to demonstrate the added value of a EuroFAANG research infrastructure here in Europe. We need to take advantage of the ongoing funded projects that we talk about today in order to really identify the needs of the users from industry, of course, but also from the scientific community and the expectation with regard to animal genetics, physiology, and health. And we need to add value and provide reciprocal benefits from using the same methods, protocols, and dissemination activities. Again, the wrong one. So in summary, our EuroFAANG research infrastructure project is contributing to addressing the need to bring together national facilities at the pan-European level for animal genetic resources, phenotyping, breeding, and animal health. And this is exactly what was identified as the gap that is to fill on the F3 roadmap. And the EuroFAANG projects builds together with the 6 Horizon 2020 projects and connects with the existing infrastructure on data management and animal agriculture at the European research landscape. And this will, at the end, lead to a hopefully successful concept to get a sustained support for having G2P research funded in Europe. And with this, I would like to thank the partners in the EuroFAANG RE project. I would like to thank the consortia of the individual EU FAANG projects. I'd like to thank the EU commissions and you for the community. And I'm really looking forward to push and sustained support of G2P research in Europe. Thank you.
Thank you, Klaus. Any questions for Klaus?
Hiroaki Taniguchi from Polish Academy of Sciences. And what kind of cell model are you planning to establish? And what kind of genome editing technology are you thinking? Only like CRISPR-Cas9 mediated method, or like base editing, or other technology are you considering?
I think that of course we need to cover what is present, what is done, what is ready, and we need to show up what developments are needed. And of course, we are open to, and we have to be open to any new advancing technologies that are coming up.
Yeah, so HeroEcho, we have one deliverable focused on using robotics and high-throughput CRISPR screens that would be interesting to you. And yeah, as Klaus mentioned, we're trying to survey the community and the labs using innovative methods using genome editing and other technologies.
Okay, thank you so much.
Anyone else? I think, should we move on given time? Yeah, okay.